Okay, so we're going to pick up where we left off in class was when I was going to go into plant versus animal cells. So let me get my screen up for you guys. Share my screen, go to here, slideshow. Okay, so let's talk about plant cells and animal cells. So I have behind me a nice little Venn diagram about the differences between prokaryotes and eukaryotes. And you guys can make some of these to help you understand the similarities and differences between plant cells and animal cells. I don't know what a plantimal is. All right. So basically, these are the components of cells that are different between plants and animals. Everything that we talked about up to this point, as far as eukaryotic cells are concerned, plant cells and animal cells have all of those things, but this is where we start to get the divergence. This is where we start to fill in the things that plants and animal cells don't share. So namely, animal cells have centrosomes and lysosomes. So zomes means bodies. So these are bodies that are the kind of the central this is what provides the structure for animal cells, the cytoskeleton. Plants don't really have a cytoskeleton, so animals have, that's one of those things that are different about them, and that really depends on the anchor point is a centrosome. Lysosomes are found in animal cells. These are organelles that are meant to break stuff down. So if we continue our city analogy, then that would be the recycling center. Plants have some things that animal cells don't have. The most obvious is a cell wall. And then of course plants have chloroplasts where photosynthesis happens. Obviously animals don't have chloroplasts, they don't do photosynthesis. We have to eat plants and other things that eat plants. And then plants have this large central vacuole that is not found in animal cells. Okay, so let's look at all of these components of a cell each in turn. So if we focus first on animal cells, we're going to talk about the centrosome. These are found near the nuclei. They're important in cell reproduction because what it does is it pulls the chromosomes toward the ends as that cell is getting ready to split. So let's take a look at the structure of these for a hot second. So there's two of them and they sit crisscrossy at 90-degree angles. And then they're made of these tubes. So there's each of these, there's 3 tubes that are partitioned together, grouped together. And then there's 9 of those partitionings per the central. So they come in microtubular triplets. And then, so their job is to attach to the chromosomes as they're duplicating and then yank them toward either end of the poles of the new cells so that each of the daughter cells are going to end up with the right amount of genetic material. Why plants don't have these, we don't really know. There's some components of the cell wall that do a similar thing for plant cells, but because animals lack plant cell walls, they have to have this other mechanism to make sure that the DNA is ending up in the right places before cell division occurs. So, okay, so that's what their job is to make sure that when it's time to divide, are your chromosomes in the right place? Lysosomes, again, this is the garbage disposal or the recycling center. You can think of it as either way. Their job is to break down other like big stuff, and then those breaks it down into the component parts, and then those component parts can go off and be used in other parts of the cells. So typically, inside the lysosome has a lower pH, which means it's more acidic. So it's easier for stuff to be broken down. And it's also important because all the cellular processes have to go on. And if the pH, if the acidity level is too high, then those cellular processes can't happen. And so we kind of need to have little compartments of this little packets of these higher higher acidic areas to do the recycling. Okay, so yeah, so there's specialized enzymes that allow that breakdown of these big macromolecules, whether that's proteins that didn't turn out quite right, whether that's invaders, viruses that are invading the cells can break those down and then use the amino acids and the nucleic acids and the carbohydrates and all that stuff in its regular cellular processes. And again, so this is only found, lysosomes are only found in animal cells. The large central vacuole in plant cells do a similar function. And so they don't need these lysosomes. Okay. So now we're going to switch over to talking about plant cells. So first thing that we're going to talk about is this cell wall. So the cell wall is on the outside of the plasma membrane. So if we're going outside to in, so we got cell wall, and then right on the inside of that is the plasma membrane. And then we have our nice cytoplasm and all the cell juice and all the components inside the cell. So what I like to do is, you can find anything on the internet, you guys. You can even find a photo of a balloon inside a cardboard box. So I like to think of the cell wall as a cardboard box. It's rigid, it provides structure, and then right inside of that is the balloon in this analogy, which represents the plasma membrane, which is, has that mosaic, it's flexible. It can expand to fill up that whole space, or it can contract like we saw under the microscopes. When we put the salt water on the Elodia leaf in the salt water, and we saw it shrink up, and that's what's happening there. So the difference between plant cell walls and bacterial cell walls basically is what they're made of. Plant cell walls are made of cellulose, and so remember that this is glucose molecules that are hooked together in this pattern that makes it hard to digest and decompose. So that's all you need to know for that. You don't need to know that it's a beta 1,4 bond, but it has this bond that makes it really rigid and strong and hard to break down. Okay, that's cellulose. It provides protection from the outside. So we think of, like when you crunch into a raw stick of celery, that crunching is coming from the mechanical breaking of the plant cells by your teeth. And that is, so that provides not only protection from things trying to get in to the plasma membrane, but it also provides structure. So if you think of, it's really easy to stack up a bunch of cardboard boxes, right? So it's the same concept here, is that the cell wall allows plants to have that structure that keeps them, and we'll talk about the vacuole in a minute and its role in providing structure, because what it does is it expands when there's a lot of water and pushes that plasma membrane up against the wall. One of the things that I used to like to do when I was a little kid was find the doorways, and we would like climb up, pushing our hands and feet against either side of the door frame and using that pressure to lift ourselves up. And then my mom would be like, get down, you're gonna break your legs, but we never did. So, but anyway, that point is that it's that pressure of the vacuole and the plasma membrane pushing against that rigid cell wall that gives plants their structure. And when plants start to dry out, and that's when they wilt, is because they don't have that structure. Okay, and then just remember that the plants, the cell wall is on the outside of the plasma membrane. So that's why this image of a balloon inside of a cardboard box is helpful to help you remember where things are located. Okay. Chloroplasts, these are basically the solar panels of the cells. So they have a double membrane. they have DNA, they have ribosomes. Again, this is another example of that endosymbiosis hypothesis where we see these organelles have the same structure and function as prokaryotic cells, and we think that it was just junk, that these things were being taken in and then incorporated into the function of this larger other cell. So chlorophyll is housed inside the chloroplasts. It's in these little, this molecule is in these stacks that we call thylakoids, and then which are then interconnected stacks of grana or 1 granum. So anyway, so just know, as long as you know that the chlorophyll is inside the chloroplast, and it's the chlorophyll that actually is capturing the energy from the light, breaking apart a water molecule in order to allow photosynthesis to occur. So this is how plants are able to make sugar, and then they can go throw that into their that sugar into their mitochondria, which then does cellular respiration and uses that sugar, breaks it down to make ATP. Okay. And then again, the central vacuole, this is kind of the convention center of the plant cell, for using that analogy. This is the huge area where we get most of the plant's volume. So in some plant cells, it can be up to 90% of the cell's volume is just this sack of water. It provides that trigger pressure that I talked about before, where you push against that rigid cell wall in order to provide you structure. And then, so this means that plants can, as long as they're well hydrated, they can increase their cell size without having to make more cytosol and produce use a bunch of energy to produce cytosol. Okay, so hopefully you guys can make a Venn diagram using this information that I have here for you. So make a Venn diagram about plant cells versus animal cells, and we can take a look at those in class on Thursday.